Hi, everyone. My name is Hope Avalone, and I am the Senior Manager of Patient Education Programs here at the Lymphoma Research Foundation. And it's my great pleasure to welcome you to our webinar, Understanding Immunotherapy for Lymphoma. During today's webinar, you'll hear from two expert speakers, and you'll have an opportunity to ask questions. You can ask those questions at any time in the Q&A box at the bottom of your screen. And we'll address as many as we can before the conclusion of the program after we complete the presentations. And as a reminder, this webinar is being recorded. Also, at the end of the program, you'll be prompted to complete a program evaluation, and we do ask that you complete this brief evaluation as we incorporate your feedback into future programming. Before we get into the presentations, I'd also like to share a few brief words about our organization. The Lymphoma Research Foundation is the nation's largest nonprofit organization that focuses specifically on lymphoma and chronic lymphocytic leukemia. Our mission is to realize the promise of science to eradicate lymphoma and, of course, to serve the community touched by this disease. And all of our work is led by our Scientific Advisory Board, which is a group of 45 leading lymphoma experts from around the country. And in addition to today's program, the Foundation has many other programs and services available for you. So we offer both in-person and virtual programs so you can continue your learning throughout the year. And I wanted to specifically highlight our upcoming lymphoma talks, which are in Los Angeles, Houston, and Minneapolis. Our lymphoma talks are weekday in-person programs with 3 breakout sessions. So one for indolent lymphomas, one on aggressive lymphomas, and then one separately on CLL, SLL. So to view the agenda and register for any of these programs, if you're in any of these areas, you can visit lymphoma.org/programs. And in addition to our programming, the Foundation also offers a wide range of support services. Our helpline has master's level trained staff who can provide individualized information on all subtypes of lymphoma, as well as on treatment options. They also can help with clinical trial navigation and connections to other resources such as financial assistance. We also offer a peer support program called the Lymphoma Support Network, and through the Lymphoma Support Network, we can connect patients and care partners with others who have been through similar experiences for emotional support. Lastly, we also have comprehensive disease guides and fact sheets, which can be ordered or downloaded for free on our website, which is lymphoma.org. And please also look for us on YouTube, where we spotlight different lymphoma subtypes and treatment options. And finally, I'd like to highlight our Spanish language programming and resources for anyone who might find resources in Spanish to be helpful or who knows someone who might find resources in Spanish helpful. I'd also like to take a brief moment to thank our program supporters. Thanks to their generosity, we're able to host programs like today's webinar. But for now, we have a wonderful program planned for you today with 2 expert speakers, Dr. Samuel Yamshan from Weill Cornell Medicine and Dr. Justin Klein from the University of Chicago. First, I am so honored to introduce our first speaker, Dr. Samuel Yamshan. Dr. Yamshan is a hematologist-oncologist at Weill Cornell Medicine, New York Presbyterian Hospital, where he's also an assistant professor of medicine. He specializes in treating patients with lymphoma using cellular therapies, including bone marrow transplantation and CAR T-cell therapy. And last but not least, he's a grantee under the Lymphoma Research Foundation Scientific Research Mentoring Program. Dr. Yamshan, we're so thrilled to have you here today, and I will now turn the talk over to you.
Thank you so much, Hope. Just going to share my screen. And excited to speak with you today, and thanks so much for the introduction. Again, I'm Sam Yamshan, and I am here at Weill Cornell Medicine, the director of our cellular therapy service, where we treat patients with lymphomas, actually now, as well as several other indications now with cellular therapies. And I'm really excited to talk with you today a bit more about kind of wrapping our head around and understanding CAR T cell therapy, which I think is a little bit different than excuse me, than some of the other treatments that you might know about already. So just briefly, these are some of my industry disclosures as well. Just wanted to disclose as well that some of the graphics and figures in this presentation were generated by the GPT-4o image generator, and you can tell me what you think about some of them. So just a quick overview of what we're going to talk about in the next 25 minutes or so. Going to start out by an overview of CAR T. What are CAR T cells and how do they work? And then most importantly, how have CAR T cells changed the paradigm for lymphoma treatment? I'm going to talk a little bit about when to consider CAR T as a treatment option for lymphoma, as well as the importance of an early consultation. Then we'll speak a little bit about managing potential treatment side effects, and then looking forward into our crystal ball a little bit about the future of CAR T cell therapy. So let's start off by kind of asking the question, what are CAR T cells? So I think kind of even before we talk about CAR T cells and what they are, I think it's one thing that's very important to understand is that our immune systems and our body normally is constantly trying to fight off invaders. And one of those types of invaders that's important is actually cancer. And so our immune systems are constantly kind of on the prowl, surveilling, looking for cells that look abnormal, and kind of throughout our life are constantly killing off cancer cells before they become anything scary. But lymphoma cells and cancer cells in general are often very smart, and so they're able to find ways to hide from the immune system. So under normal circumstances, here on the left, our immune system actually requires specific proteins on the surface of cells in order to recognize them as cancer cells or as foreign cells and to be able to attack them. And so what lymphoma cells are actually often able to do is they're actually able to just stop expressing that protein, or they're able to find other ways to shut down T cells. And so what we're actually now able to do with chimeric antigen receptors is we're able to take T cells out of people's bodies, and we're able to reprogram them by putting in a new protein onto the surface of those T cells that we call the chimeric antigen receptor. And essentially what that allows us to do is to allow these T cells to be able to see the lymphoma cell again and to attack it even when it's trying to hide. And what's critical about that chimeric antigen receptor is that it allows us to target specific things, and that actually has implications beyond lymphoma because it means that we can actually choose whatever target that we want. And so this is part of what makes CAR T cells so exciting. But as you noticed, that receptor was actually only part of the story. And as it turns out, at the very beginning, when we first started doing CAR T cells a couple of decades ago, that wasn't enough for the T cells to be able to see the lymphoma cells. And so the addition of what we call a co-stimulatory domain, which sometimes I like to colloquially refer to as the gas pedal, is actually very necessary for those CAR T cells to activate. And so once the CAR T cell detects the lymphoma cell, it actually then kind of kicks in the gas, and those CAR T cells turn on and activate and go after the lymphoma very aggressively. So the manufacturing of CAR T cells is actually a multi-step process. And so the way that we do it, as I mentioned, is we're actually able to, the first step is to take T cells out of people's bodies. And typically what we do is we use either two IVs, one in one arm and one in the other arm, or in people where they might not have great veins, we sometimes are able to put in a special kind of IV either in the chest or in the neck. And what we do is we actually remove some of the blood from people's bodies, and we put them through a filtering machine. This process is called apheresis. And basically what we do is we actually filter out some of those immune system cells, and then we return the rest of the blood back to the body. And so people aren't actually losing blood. We're just filtering the blood for some of those immune system cells. So then, once those T cells are collected, we're able to then separate them from the rest of those immune system cells, and then we send them off to the company. And the way that I typically refer to this is I call this step boot camp, where those cells then go off to the company and they're trained over the course of weeks to be able to recognize and kill the lymphoma cells. And from a scientific standpoint, what we're actually doing during that time is inserting the genes for those new proteins onto the surface of the lymphoma cell. Many of the ones that are available commercially actually use a virus to do that step. And then once we've done that, we then grow those cells in a lab until there are millions and millions of them. And then we get a call from the company that the cells are ready to go. We then bring people back to our office and we give them a very light dose of chemotherapy for 2 to 3 days. The purpose of that chemotherapy is actually not to do anything against the lymphoma itself, but actually to make room for those new T cells to come in and, kind of establish themselves in the body. And we call that process, you may hear it referred to as lymphodepletion. Then people get 2 days of rest, and then we infuse the cells into the bloodstream. Despite the long and process, this is actually the most anticlimactic step. The cells drip in either through a bag over about 20 to 30 minutes, or are pushed in through a syringe. And once they're in, because these T cells are actually parts of people's body, they're actually part of patients' own immune system. Once they're in the bloodstream, they are able to expand quickly and get to work at killing lymphoma. And I sometimes kind of refer to them after that boot camp step. They're now the special forces that are able to kind of seek out and kill the lymphoma. But I think even more so than just the manufacturing process alone, the process of coming from referral to actually receipt and then recovery from the CAR T cells is actually requires many different people, and it really does take a village to deliver CAR T cells. So typically the steps to getting to CAR T actually come well before that manufacturing step, and so a doctor will refer a patient to a CAR T cell specialist. Although many cases, I think increasingly around the country, many of us lymphoma doctors also kind of double as CAR T cell specialists. So it's possible that you may not need a referral. Typically, there's some medical workup to make sure that CAR T cells are going to be safe. There is sometimes a process of obtaining insurance approval if needed. Then that boot camp step happens. And one important thing is that during that process of the cell manufacturing, if needed, many patients actually receive what we call bridging therapy, which is treatment to kind of keep the lymphoma at bay while we're waiting for the T cells to manufacture. And so in many cases, we do some sort of therapy while we wait, be it chemotherapy or radiation or sometimes something else. Then, as we noted, the lymphodepleting chemotherapy portion to make room for the cells, followed by that CAR T infusion. And as I said, once the T cells are in, they are doing their job, and typically for that first week after the CAR T cells, people are actually monitored daily to monitor for side effects. Sometimes in some centers that actually requires a hospitalization for at least 7 days. At other centers, we're actually now able to do this outpatient. And then, excuse me, after those first initial days of monitoring, patients are also required to stay close to the medical center for a couple of weeks after treatment, and are not allowed to drive a car during those first 2 weeks. And that was actually recently changed by the FDA just a couple of weeks ago. It used to be much longer, so we're all very grateful for that change. And then after that initial treatment, generally speaking, people either continue to follow with their CAR T doctor At our center, I would say for the first few weeks after CAR T, we see people at bare minimum weekly, and sometimes more frequently if people are having side effects, which we'll get to. But after that initial period, especially for people who are coming from far away for CAR T cell therapy, they are able to return back to their general oncologist or lymphoma doctor and continue treatment from there. And so, as you can see, there are many different steps in this process that are required, and it really does take a village. So I'm going to talk a little bit about, this is a very long, drawn out and complicated process. But I think part of the reason why we do it is because it really has been such a paradigm shift for lymphoma treatment. So CAR T was first approved in diffuse large B-cell lymphoma. And, as some of you may know, for patients who are diagnosed with diffuse large B-cell lymphoma, actually many are cured with frontline chemotherapy. But when the lymphoma comes back after the chemotherapy, the outcomes have historically, the lymphoma has been a little bit more difficult to treat. And so we, over the years, have had many different ways in which we've tried to treat lymphoma, but many of them have involved chemotherapy-based approaches. And so CAR T kind of grew out of an attempt to attack the lymphoma at a completely different angle. And so currently there are 3 different approved FDA-approved CAR T cell products in diffuse large B-cell lymphoma, which, as I mentioned, was the first approval in lymphoma. And currently, these 3 products are approved as the 3rd treatment or greater, so people who have had at least 2 prior therapies. All 3 are very similar in that they target the same target on the surface of the lymphoma cells called CD19, and they differ very slightly in other ways. So axicabtagene ciloleucel, axicel, or Yescarta has a different gas pedal, the CD28, than the other 2, which have 41BB as their costimulatory domain or gas pedal. Tisagenlecleucel, Tisacel, or Kymriah, and lysocabtagene maraleucel, Lysacel, or Breyanzi are very, very similar in that they both have that 41BB gas pedal, but they differ slightly in that there's an additional manufacturing step at the end to adjust the T-cell ratio with lysocell. And all 3 products, I think very importantly, produce durable remissions in a subset of patients with diffuse large B-cell lymphoma. And as I always emphasize to my patients who are receiving CAR T-cell therapy for diffuse large B-cell lymphoma, the goal with CAR T is very much cure. And I think at this point now we have CAR T has been approved for 7 years with diffuse large B-cell lymphoma. CAR T I very much think of as a treatment that can and does cure many patients with DLBCL. And so because CAR T cells were so effective in treating lymphoma that had come back after at least 2 treatments, the question kind of became, well, wait a minute, if it does so well after multiple treatments, can it do better earlier on? And so 3 different CAR T cell trials were done comparing getting CAR T as the second treatment versus getting getting chemotherapy again a second time, and then doing CAR T cells afterward. And what they found was that there was a pretty massive benefit in how long it took for the lymphoma to come back in getting CAR T cells versus chemotherapy. And so at this point, CAR T cells have very well established themselves when the CAR T cells, when the lymphoma comes back early for diffuse large B-cell lymphoma. And I think importantly, studies have also shown that CAR T cells, in addition to how long it takes for the lymphoma to come back, we've also shown that for axicell patients actually live longer, which is really just the gold standard. And so CAR T cells, I think at this point have just been a total paradigm shift in how we think about treating diffuse large B-cell lymphoma. And for the sake of time, I'm not going to get into all of the data for some of the others, but kind of coming on the back of the incredible efficacy in diffuse large B-cell lymphoma, we now have FDA-approved multiple FDA-approved CAR T-cell products in follicular lymphoma and mantle cell lymphoma as well. And actually, I didn't include it on here because at our center, we tend to think of CLL as more of a leukemia, but CAR Ts are actually now also approved in CLL. And so just again, like a total paradigm shift in how we think about treating patients with lymphoma. So when should we consider CAR T? So just as I did want to provide a quick list of some of the FDA-approved indications for CAR T cells and B-cell lymphomas, just because I think it could be helpful for some people on the webinar. So in diffuse large B-cell lymphoma, there are several ways in which CAR T cells are approved, but for really, the way I think about it is for anyone for whom the lymphoma has come back after the initial chemotherapy, I do think it's worth at least thinking about doing CAR T cell therapy. In follicular lymphoma, it's currently approved after at least 2 prior treatments, and in mantle cell lymphoma after 1. And so, but one thing that I do want to emphasize is that it's kind of never too early to start thinking about CAR T cells. And so, and I think this is for a few reasons. So one is that the process of CAR T does take time, as well as a referral to a specialized center. And so if for people who are currently being treated at a center that doesn't offer CAR T, I think it's always a good idea to kind of get established and meet someone from a CAR T center so that if later on this is something that it's decided that we should be able to move quickly. And as we noticed in some of the earlier slides, the process of CAR T is a multi-step process, and so can be lengthy. And so it's always kind of a good idea to be established relatively early. One thing that we try to do at our center is I actually get 90+% of my referrals by text message. And so kind of establishing a relationship with a CAR T center is, I think, makes this process a lot easier. Another reason I think to consider CAR T early is because we do have some data that CAR T may be both more effective and less toxic the earlier on in people's treatment. Not that we don't do it later, but that it may be more effective and less toxic early. And I think importantly, it's always a good idea to be in communication with a CAR T center because certain therapies can actually increase the chances that they may not be able to manufacture the CAR T as well. And so it's always a good idea for someone who's treating the lymphoma, if they think that CAR T could be something that we think about in the future, to kind of think about some of those therapies potentially. And so we do typically recommend for people to come meet a CAR T doctor early, even if it's not something that we're thinking about right in this moment. So a little bit about some of the toxicities of CAR T. So CAR T cells can have some unique side effects. And the way that I think about the side effects of CAR T is that, remember that we talked in the beginning that, our immune system is constantly fighting invaders. And the CAR T is treating the lymphoma like an invader, just like it would treat any other invader, like, a bacteria or a virus. And so the way that I think about the side effects of CAR T cell therapy is that, for example, when you get the flu, the reason why you feel so uncomfortable when you get the flu, the fevers, the body aches, is actually largely not because of the flu itself, but because of your immune system that's responding to the flu. And so I think about the side effects of CAR T cells much in the same way, that the body is creating inflammation by going after the lymphoma, just like it would go after any other invader. And because of that, many of the symptoms of CAR T cell therapy are just like having a bad infection, and actually can sometimes even be difficult to differentiate. So, for example, fever is extremely common. For some of the products, actually, the majority of people get some form of fever, and that fever can be sometimes quite high, 102, 103 degrees. Just like with a bad flu, that fever is often associated with body aches and fatigue. And sometimes, just like with a bad flu, that fever can be associated with racing heartbeat, with shortness of breath, with low blood pressure, which is a big part of why we do these treatments with very close monitoring, because unlike with an infection where we can just say give antibiotics, instead we have to kind of target the inflammation itself. And so we have a lot of tools in our toolbox to kind of tone down the inflammation and calm down those symptoms so that they don't become severe. And so if those symptoms occur, even for many centers that are giving CAR T in the clinic, we generally do treat these symptoms in the hospital. Another strange symptom that can sometimes occur is we refer to as neurotoxicity. And this, I think, is a little more poorly understood than the general inflammation. But in the same way that sometimes when people get the flu, they can get delirious, that can sometimes happen with the CAR T too. This can often be disconcerting for patients and especially for their families. And so it's something that we actually monitor for very closely and kind of intervene very quickly to calm down. We actually often give patients a quiz in the hospital every few hours to try to make sure that we're catching it early and intervening. And all of these symptoms, these fevers, the confusion symptoms, if we intervene early and aggressively, are generally able to get under control pretty quickly and are generally temporary. Long term, there is an increase of infection after CAR T cells, and that I think is related both to the chemotherapy as well as the fact that in the process of getting rid of the lymphoma, we're also getting rid of some of the normal B cells. And so people can often have low antibody levels for a very long time after the CAR T. And for people who get recurrent infections, we actually often are able to give people antibody replacements to prevent some of these things. But this is something that we definitely watch for long term. Low blood counts can often happen after CAR T cells. I would say the majority of people, this occurs early, and I know that this is a very busy, complicated slide, but this is just kind of to highlight the fact that the reasons for low blood counts after CAR T, there can be many, many different reasons, and so it's really important to have a CAR T doctor who is experienced and is able to kind of evaluate for many of these different kinds of low blood counts after CAR T. But many times these low blood counts are manageable after the CAR T once we're able to identify the reason why they're occurring. And I think kind of to highlight, the CAR T must be given in a specialized center. As I mentioned, the FDA does require that people will be seen daily at the center for at least 7 days and then be within 2 hours for 14 and no driving for 14 days. And these short-term side effects, especially the fevers, the confusion, do require expertise as well as the availability of some of those specialized anti-inflammatory drugs. But the long-term side effects can often be co-managed, and so people don't have to stay at the CAR T center long-term necessarily. So just quickly looking forward to some of the future of CAR T cells. So how do we improve upon CAR T cells? And that the improvements in CAR T can come in many different ways. One way might be bringing CAR T earlier in treatment. As I mentioned, we have data that getting CAR T earlier, seems to be both more effective and potentially less toxic. So that's a potential consideration. We also are, very interested in finding ways to improve the efficacy of CAR T cells in different ways, and either by trying to find new targets or reducing the T cells getting tired out over time, as well as ways to improve the safety of CAR T. So just to highlight a couple of things, so there are several clinical trials bringing CAR T earlier in the treatment paradigm for lymphoma, and so in diffuse large B-cell lymphoma, we actually have a study that is looking at giving CAR T as the first treatment for high-risk diffuse large B-cell lymphoma and trying to avoid some of the toxicity of getting several rounds of chemotherapy, so trying to replace chemo with CAR T. And similar studies are ongoing in follicular lymphoma. But also, I think, even for the lines of treatment that we already have, trying to find ways to um, mitigate some of the ways in which lymphoma is able to escape CAR T cells. And so kind of finding ways to make the CAR T cells become less exhaustible and ways to make the lymphoma cell become more targeted. So just a quick list. I mean, there's literally dozens and dozens of studies out there trying to improve CAR T cells, but just to highlight some of these strategies, we think that potentially manufacturing cells faster and getting the cells in fresh rather than frozen may inhibit the T cells from getting as exhausted. There's actually some studies using T cells from donors, which we think may be healthier since they've never been exposed to chemotherapy. Many different CAR T cell products that are both targeting new targets as well as multiple targets at the same time, so that if the lymphoma hides its stripes, it can still attack at a new angle. We have studies here using other medicines alongside CAR T to supercharge the T cells and prevent them from becoming exhausted. And also, of course, developing new prediction models so that we can predict who may need additional therapy. And then in terms of safety, there are several ways in which we're trying to improve safety. Some ways are by potentially changing that gas pedal protein to make that, hitting the gas a little less intense. Developing CAR Ts with on and off switches so that we're able to tone things down if the toxicity gets too intense. And then using medicines to prevent some of these toxicities from occurring. An ounce of prevention is worth a pound of cure, as well as honing some of those strategies for bridging therapy to control some of the inflammation prior to CAR T. So to sum up, because I want to make sure that Dr. Klein and has time and also we're able to get to Q&A. CAR T is a very effective treatment for B-cell lymphoma. Early discussion of CAR T can make a difference, and I do encourage people to ask their lymphoma doctor about the potential of an early CAR T consult. CAR T can have some strange toxicities that require management with a specialized center, and we have many different strategies that are both trying to improve the outcomes and the safety of CAR T. So thank you for your time, and I do just also wanted to share my email here. I know I actually think some of my patients are on this webinar, and they can attest that I do respond directly to email from patients. And so if you want to reach out and contact me with any questions, please feel free to reach out anytime. And thank you for your time, and looking forward to talking at Q&A.
Thank you, Dr. Yamshan, for getting us started with your presentation and for sharing your contact information. I think that's really helpful. And I'm now very thrilled to introduce our next speaker, Dr. Justin Klein. Dr. Klein is a hematologist oncologist at the University of Chicago, where he is the director of the lymphoma program and a professor of medicine. He has expertise in the development and use of immunotherapy, and his lab research is focused on improving the effectiveness of immune-based treatments for people with lymphoma. He's participated in a number of our patient education programs, and so we're happy to have him back today. Dr. Klein, I'll now turn the talk over to you. Thanks so much.
Thanks for having me, Hope. And Sam, thanks for a great talk on CAR T cells. Okay, so over the next 15 or 20 minutes, I'll talk about some other therapies that depend on the immune system, specifically antibody drug conjugates and bispecific antibodies, which are broadly used in lymphoma these days. Here's a brief outline. I'll talk about ADCs and bispecific antibodies in general, how they work, where they're used in lymphoma treatment, some of the side effects that people may experience when being treated with ADCs or bispecific antibodies, and then very briefly, looking for how are these drugs and therapies going to be used down the road. And so to understand antibody drug conjugates and bispecific antibodies, you need to know what an antibody is. And antibodies are basically proteins that are produced by immune cells called B cells. And interestingly, each of our B cells generates a single unique antibody. And of course, that helps to create diversity in our immune system that allows our immune system to recognize a broad array of pathogens like bacteria and viruses and so on. And the antibody in cartoon structure sort of looks like a Y. Sort of the ends of the Y are what binds to the target. The fancy word for that is called the antigen. And the back end of the antibody is what signals to other cells in the immune system that an antibody is bound to a bacteria or to an infected cell, for example. And so these antibodies are very important in helping our bodies prevent and fight infections. And this is sort of the business of antibodies. Basically, the antibody, these Y structures, they stick to specific targets, and those targets could be proteins on the surface of an infected cell or a cancer cell, for example. And then once bound to their targets, again, that signals other cells in the immune system, such as cells called macrophages or natural killer cells, to recognize those cells as being abnormal and then to get rid of them, basically. Antibodies can also stimulate immune proteins called complement proteins to attach to the cell, leading to its death. And so this is basically how antibodies help us fight infections. Now, our blood contains a huge variety of antibodies, each with a different specificity, each able to bind to a different target. And each of these antibodies has been produced by a unique B cell. And these are what are called polyclonal antibodies, meaning antibodies of broad specificity. And although these are important for the diversity of our immune system, they're not very useful therapeutically. Now, if we could identify a specific antibody, a single antibody produced by a single B cell that recognizes a target of interest, say a target on the surface of a lymphoma cell, those antibodies are known as monoclonal antibodies. They're all exactly the same. They have the same target. And if we can identify a monoclonal antibody specific for a target, say on a cancer cell or a lymphoma cell, now that's something that can be used therapeutically. And it turns out that many therapies used in lymphoma today are based on monoclonal antibodies, probably the best known of which is a monoclonal antibody that many of you are probably familiar with called rituximab or Rituxan. And this is an antibody that targets a protein on the surface of B cells, lymphoma B cells, and as well as healthy B cells. But when rituximab antibody is bound to the B cell, or when it sticks to the B cell, it activates what are called effector responses, either the activation of those immune proteins called complement, or effector immune cells like macrophages and or NK cells that ultimately kill the B cell. So let's talk about antibody conjugates, antibody drug conjugates, or ADCs. ADCs are basically monoclonal antibodies with a bit of junk in the trunk, so to speak. The antibody itself, this Y structure, is linked to a payload, which is code for chemotherapy, essentially. And so the antibody portion of the ADC binds to the cancer cell. That structure is brought inside the cell in a structure called an endosome. The low pH or the acidic pH in that endosome releases the payload, and ultimately the cancer cell dies. So ADCs are basically clever tools for targeting delivery of chemotherapy more specifically to cancer cells. And this somewhat complicated cartoon sort of gives you the sense of how these ADCs work. Basically, the antibody drug conjugate on the upper left binds to its little purple target or antigen. The structure is brought inside the cell into the endosome, where the payload or the chemotherapy is cleaved and released into the cell. And based on how that chemotherapy works, it can either integrate into the DNA of the cell, preventing it from duplicating itself, which is a process that's required for a cell to divide, or it can disrupt these structures called microtubules, which are structures that are literally important for one cell to divide into two cells. And again, it's these ADCs are, the efficacy or the effectiveness is dependent on chemotherapy, but the chemotherapy is more, the delivery is more streamlined to target cancer cells as opposed to any dividing cell. And so how do we use ADCs to treat lymphoma today? Well, the first ADC that was approved in lymphoma was one called brentuximab vedotin. We call it BV for short. This targets a surface protein on lymphoma cells called CD30. This was initially approved to treat Hodgkin lymphoma, and more recently has also been extended to be used in relapsed large B-cell non-Hodgkin lymphomas. And it can be administered alone in the case of Hodgkin lymphoma or in combination with other chemotherapies or immunotherapies. The second major ADC used in lymphoma is called polatuzumab vedotin. We call it POLA for short. This is an ADC that targets a different cell surface protein on B cells. It's called CD79B. This one is mostly used to treat diffuse large B-cell lymphoma, and it's often combined again with other chemotherapies or immunotherapies to enhance its effectiveness. And then the third is an ADC called loncastuximab tesirine. These all have fabulously complicated names. We call it Lonca for short. And this is an ADC that targets the same target as most CAR T cells, which is another protein on B cells and lymphoma cells called CD19. And this is mostly used to treat large B cell lymphoma and more recently, perhaps follicular lymphoma. And this is a drug that's typically administered by itself or in some cases with Rituxan. I'm just going to highlight a couple major advances in the field of ADC therapy. The first is in large B-cell lymphoma, where we've used for many, many years a cocktail of chemotherapies, Rituxan, and corticosteroid called prednisone. The cocktail is called RCHOP. It's been around since before I started in the business. And many other treatments have been tested against RCHOP and have failed to improve outcomes. However, when the O drug in CHOP, which is a chemo called Oncovin, was replaced with polatuzumab, it turns out that patients that received polatuzumab with rituximab, Cytoxan, doxorubicin, and prednisone had better outcomes than people that received R-CHOP. And so this has become a new standard of care for the frontline or initial treatment of people who are diagnosed with diffuse large B-cell lymphoma. So this is a huge advance, and this is really only a few years old now. Another example is in Hodgkin lymphoma, where that initial ADC, the BV or brentuximab vedotin, was combined with some chemotherapy medicines and compared to the standard of care. And with several years of follow-up, was the first cocktail to really show improved overall survival in Hodgkin lymphoma. And so you can see that antibody drug conjugates initially approved in relapsed lymphomas have been so effective they've made their way into being incorporated into initial therapy for both Hodgkin and some non-Hodgkin lymphomas. Now, although ADCs target chemotherapy to lymphoma cells, they do have some side effects. I've listed a few of the key side effects here. Brentuximab vedotin can cause things like fatigue or nausea. The major side effect that we watch out for is neuropathy, which is damage to the nerve endings in the fingers or toes that can cause numbness or pain. It can affect the blood counts, although not markedly so, and it can cause inflammation of the liver or pancreas less commonly. Polatuzumab, I've found personally, is very well tolerated, but can cause fatigue or some diarrhea, can also cause some neuropathy and low blood counts as well. And finally, loncastuximab is, in my experience, pretty well tolerated, but probably the one that's a bit roughest among the three. It has some unique side effects, including swelling in the body, skin rash, and sun sensitivity. It also can inflame the liver, causing sometimes some nausea and low blood counts as well. So these medicines, I think, in general, are better tolerated than conventional chemotherapies that we use to treat lymphomas, but they do have some side effects that we have to watch out for. And then moving on to bispecific antibodies, and these are also known as bispecific T-cell engagers or BiTEs, or you might see them called T-cell engagers. These are antibodies that have been modified such that one arm of the Y is able to recognize a protein on T cells called CD3, And the other arm on the Y recognizes a target on cancer cells, in our case, lymphoma cells. And so it sort of juxtaposes a T cell with a tumor cell or a lymphoma cell. And simultaneously, the binding of this bispecific antibody to that CD3 protein on the T cell triggers its activation, allowing it to secrete proteins that lead to the death of the lymphoma cell. So it's a clever way of redirecting the specificity of T cells, which, like B cells, have their own unique receptors, but it sort of redirects their specificity to be able to recognize and kill tumor cells. Now, bispecific antibodies, many of them have been developed. They have different structures, which are not super important for us to understand. But there are many in development for lymphoma as well as for other cancers. Probably the most common one that's used in the clinic today are the two at the bottom right here. And the first bispecific antibody or BiTE that was approved to treat a blood cancer was one called blinatumomab. And this is a bispecific antibody that activates T cells through CD3 and also binds to CD19 on, in this case, leukemia cells. And this was a number of years ago approved to treat relapsed B-cell acute lymphoblastic leukemia. And led to pretty impressive response rates. Those responses tended not to be terribly durable. So in this case, patients often were recommended to undergo donor stem cell transplantation. But the development of blinatumomab identified some of the side effects that can happen with bispecific antibodies, which can be shared with CAR T cells, including cytokine release syndrome, as well as neurological toxicities that Sam mentioned in his talk. So how do we use bispecific antibodies to treat lymphoma? One thing that was interesting to me is that despite the effectiveness of blinatumomab against B-cell leukemias, its development in against B-cell lymphomas was I thought delayed, but in the last 3 or 4 years, I think has really come to fruition. There are a couple of bispecific antibodies that we use probably most widely in lymphoma. One is called epcoritamab. Another one is called glofitamab. And these are bispecific antibodies that, again, target CD3 on the T cell, and again, the CD20 protein on B cells. When they were tested in folks with relapsed large B-cell lymphomas, I would say that their effectiveness was relatively similar. You can see response rates of around 50 to 60%, and that's the percentage of people who were treated with these bispecific antibodies whose tumors shrunk significantly. Importantly, complete responses where a PET scan goes entirely blank were seen in about 40% of people. So that's pretty exciting. You know, particularly for people who achieve a complete response with these bispecific antibodies, those responses tend to be pretty durable, potentially even as durable as those that are seen with CAR T cells. So these are really effective therapies, but they're most effective in people who respond deeply, who have complete responses. Like antibody drug conjugates, bispecific antibodies certainly have some side effects. I won't go through them with regard to each of the specific bispecific antibodies that are FDA approved to treat lymphoma because it's largely a class effect. Each of the bispecific antibodies has similar side effects. You might see them with different rates in different bispecific antibodies, but overall they're pretty much the same. The first being cytokine release syndrome, which includes symptoms like fever, low blood pressure. I think Sam mentioned flu-like symptoms, which I think is a good description. The cytokine release syndrome that occurs with bispecific antibodies tends in general to be less exciting than what we see with CAR T cells. You can also rarely see neurological side effects with bispecific antibodies. We also see low blood counts and in particular infections, which I think is probably the biggest issue with bispecific antibodies today. to mitigate some of those side effects, particularly cytokine release syndrome, the initial cycle of bispecific antibody treatment is typically given in a step-up dosing fashion. So you start with a very small dose, then the next week a higher dose, and so on. And most of the side effects, particularly CRS and neurotoxicity, when we see it, occurs really for the most part during cycle 1, maybe cycle 2. After that, it's pretty uncommon to see either of those side effects. And I think it's important that folks be taking preventative antibiotics to reduce their chance of infections while on these treatments. And so I think probably the biggest future direction with bispecific antibodies is really combinations with other therapies. Unlike CAR T cells, which are a little bit more challenging to combine with other treatments, I think bispecific antibodies and their general pretty good tolerability lends themselves to combinations. And we've seen that recently, for example, in a couple of studies that have paired the bispecific antibodies glofitamab or epcoritamab with chemotherapy, gemcitabine and oxaliplatin, which we know is GemOx, and show pretty significant effects, and which was somewhat surprising to me, I guess I'd have to say. And then there are a bunch of clinical trials that are testing bispecific antibodies with initial therapies for, say, large B-cell lymphoma. Whether they're going to improve the outcomes that we have today, I think remains to be seen. But I think we're all hopeful that that might be the case. Also, there are combinations of bispecific antibodies with antibody drug conjugates, which I think are even perhaps more exciting because ADCs are, in general, again, better tolerated than conventional chemotherapy drugs. And here we have bispecific antibodies like mosunetuzumab or epcoritamab combined with polatuzumab. And so when thinking about advantages versus disadvantages or pros and cons of bispecific antibodies versus CAR T cells, I thought I'd make sort of a laundry list here. Certainly, one advantage of bispecific antibodies is that they're readily available. We can simply prescribe them. We can start them quickly. Pretty much anyone can get them. Also, in general, when compared to CAR T cells, bispecific antibodies are a bit safer. They don't require conditioning or lymphodepleting chemotherapy before their administration, which is nice. On the other hand, while CAR T cell therapy is sort of a one and done type of treatment, bispecific antibodies are often given over many cycles, sometimes as indefinitely as long as they're working. CAR T cells are also require a blood cell collection or leukapheresis and manufacturing time, which is sometimes challenging because people have lymphomas that are growing quickly and we need to control them while we wait for the CAR T cells to be manufactured. This, of course, is not the case with bispecific antibodies. These are sort of ready to go off the shelf therapies. And then, I think when comparing effectiveness, although bispecific antibodies can be associated with very durable remissions in a subset of people who receive them, my sense is that in general, they're a bit less effective than CAR T cell therapy. So if I had a person who could either receive a bispecific antibody or a CAR T cells, I would choose CAR T cells, again, generally speaking. And so to conclude, antibody drug conjugates and bispecific antibodies are both treatments based on monoclonal antibodies. ADCs deliver chemotherapy to lymphoma cells in a targeted manner, which makes them a bit safer. They're broadly used in both Hodgkin and non-Hodgkin lymphomas, including as part of initial treatments, which is really impressive and demonstrates how effective these drugs are. Bispecific antibodies work differently. They sort of juxtapose T cells and lymphoma cells and simultaneously trigger T cell activation, which allows them to kill the lymphoma cells. How clever. Bispecific antibodies, the ones that we use in the clinic, are what we call CD3, but that's a typographical error there. Should be CD3 by CD20 targeting, and are used to treat relapsed follicular lymphoma and large B-cell lymphoma, but I suspect their use will be broadened to include other non-Hodgkin lymphomas as time goes on. As I mentioned, bispecific antibodies can induce durable responses in a subset of people who receive them, but when compared with CAR T cells, I think bispecific antibodies are a bit safer but perhaps less effective. And so generally speaking, when I have the choice, I will choose CAR T cells. All right, I'll stop there. There's my email address, and we're happy to address any questions you guys might have.
For your excellent presentation, and again for offering your contact information there. We're now going to jump into some questions. We have plenty of time for those. I've been taking a look. We've got some great questions, and I'm going to aim to combine questions where I can. So starting off with a CAR T question for Dr. Yamshan. I actually have two questions here. First, what contributes to manufacturing failure of T cells, and how common is it? And next, how do the reinserted T cells kill lymphoma cells as compared to normal T cells?
Yeah, so those are both great questions. So the first question, just to recap, was what contributes to CAR T manufacturing failure, and how common is that? So I'll actually answer the second part first. Thankfully, it's relatively uncommon. So, and part of that has been that we've gotten better at manufacturing CAR Ts over time, but also what actually kind of the rules that the FDA would allow have actually gotten a little more lenient. And so a few years ago, I would say it was, depending on the product, it was around like high 80s, low 90%. And now it's well upwards of 90% manufacturing success. We still do get like a couple of every year who have CAR T cell products that we will call out of spec, which means out of specifications. So the FDA has a number of parameters that they measure in the CAR T cell product. But actually, that doesn't always mean that you can't give the cells. And so many of the companies actually have what they call an expanded access protocol, which basically means that the cells, for whatever reason, don't quite meet the FDA specifications, but we think that there's a good chance that they'll still be effective. And then we're able to give them on one of these expanded access protocols. And so I would actually say, knock on wood, but I can count on one hand the number of times where I've wanted to do CAR T cells and was not able to because I wasn't able to get a viable product. It's very, very uncommon that you're just not able to get a product. So the other question, the other part of the question, I guess, was what contributes to that. So the number of prior treatments we know does contribute in some way to that. And in particular, there are some therapies that we know to be toxic to T cells. And so one of those is a medicine called bendamustine, which we know is toxic to T cells in the short term. Bendamustine is actually a very effective treatment for lymphoma on its own, especially in the indolent lymphomas. And there are many, many patients who have gotten bendamustine who were still able to perfectly collect CAR T cells, but usually the amount of time from the bendamustine matters. And so when we're thinking about CAR T cells in the short to medium term, I often do like talk to the lymphoma doctor and say, hey, like what treatment are you thinking about doing? Maybe try to avoid this one if we think we're going to be moving to CAR T in the next, say, 3 to 6 months. And then the other question, remind me, I'm sorry.
No, it's okay. How do the reinserted T cells kill lymphoma cells as compared to normal T cells?
Yeah, so great question. So the normal T cells are constantly trying to kill the lymphoma cells, but the lymphoma cells are both very good at hiding from them and also in kind of like I don't know if you've ever played Mario Kart, but like kind of throwing bananas in the way of the T cells at getting to the lymphoma. And so what the CAR T does that's unique relative to the regular T cells are, number one, they have this protein on their surface that's able to recognize the lymphoma cell even when it's kind of trying to hide from the regular T cells. And then the other thing is that co-stimulatory domain, that gas pedal protein that kind of like ramps them up. I do think that long term, the regular T cells, I think we are increasingly understanding do play a role in keeping the lymphoma at bay. And so helping out with general T cell exhaustion, I do think is an important thing. And I do think that they kind of do, especially after those initial couple weeks, do kind of work in tandem to keep the lymphoma cells under control. But those are kind of the ways in which they differ in that initial period.
Thank you. And this next question's for Dr. Klein. Are antibody drug conjugates currently used as a frontline or first line therapy?
Yeah, so there's a couple of instances where antibody drug conjugates are used as frontline as part of frontline treatment for lymphomas. The one place is in diffuse large B-cell lymphoma, which of course is the most common type of non-Hodgkin lymphoma that we see. This is sort of an aggressive disease that's typically treated with a combination or a cocktail of different medicines. And into that cocktail, a recently published, well, relatively recently published study called Polarix tested the inclusion of an antibody drug conjugate called polatuzumab or Pola with several chemotherapies and a steroid drug and Rituxan, and showed it to be a bit superior to what we had been using for the past couple of decades. And so including polatuzumab with what we call chemoimmunotherapy has become a new standard treatment for large B-cell lymphoma. The other instance is in Hodgkin lymphoma, where an antibody drug conjugate called brentuximab vedotin, also when combined with some other chemotherapies, was also shown to be superior to the standard chemo cocktail that was used to treat Hodgkin lymphoma initial treatment. That cocktail of drugs that includes brentuximab vedotin has, fortunately, I guess you could say, already been surpassed by a different immunotherapy that we haven't talked about the mechanism of today. But that would be another instance where you could incorporate an antibody drug conjugate into initial therapy for lymphoma.
Thank you.
Mhm.
Dr. Yamshan, if someone with DLBCL has CAR T cell therapy and it's successful for years, but their lymphoma returns, can CAR T be repeated?
So that's a great question. So what I would say about that is right now, commercially, no, but there are many, 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 as I mentioned, different CAR T cells that are currently in clinical trials that are available in the post-CAR T setting. And my guess is the vast majority of CAR T centers around the country have at least one of those clinical trials available for post CAR T, like if a lymphoma comes back after CAR T, getting some sort of investigational CAR T. And so many of those trials will differ. As I mentioned, some will try to target a different spot on the lymphoma cell. Some will try to add on what we call an armored CAR, which is kind of an additional gas pedal to the CAR T. A lot of different allogeneic using cells from a donor, a lot of different strategies out there. But I actually think that at this point, at most academic centers, there is some sort of CAR after CAR available. But also to many of the treatments that Dr. Klein was discussing are actually now available as standard of care after CAR T, and the bispecifics in particular have kind of been studied specifically after CAR T cells and are quite effective. And so there are a lot of treatment options, both on clinical trials and in standard of care for CAR T, but currently the commercial CAR Ts you can't do again.
Thank you. Dr. Klein, can you talk a little bit about the use of odronextamab? I hope I'm saying that right. As a bridging defense for relapsed DLBCL, this person notes that it was tested in some trials in Europe.
Yeah, so odronextamab is another of the CD3 by CD20 bispecific antibodies. This is like glofitamab and epcoritamab, which I would call its closest cousins, if you will. Is certainly an effective treatment for relapsed large B-cell lymphoma, as well as other lymphomas potentially. It's, to my knowledge, not yet approved for use in the United States, although my sense is that its effectiveness is largely similar to glofitamab and epcoritamab. And so while we're not using odronextamab yet so much in the clinic, that may happen in the future here in the U.S. If you're somebody who lives in the United States, you certainly, if needed, could be treated with the other bispecific antibodies, specifically in large cell lymphoma, glofitamab or epcoritamab.
Thank you. And Dr. Yamshan, someone wrote here, and I know we've received a few questions. Is there ever a reason not to do CAR T-cell therapy? And I know we typically get a lot of questions around age and comorbidities.
Yeah. Yeah. And so I actually think that this is one of the most important questions in CAR T is when to do it and when not to do it. And I actually think that my guess is if you asked a bunch of lymphoma doctors around the country, you might get different answers depending on who you asked. Part of the way that I think about it, kind of to some of the points that Dr. Klein was making as well, which is that I think CAR T is probably the most effective therapy that we have, especially in the aggressive lymphomas. And so, and as I mentioned, I think we have very strong evidence that it is a curative therapy. And so because of that, I actually have a very, very high threshold not to give CAR T, meaning that I have to have a very, very, very good reason not to. And so reasons that might be would be someone who has very severe medical comorbidities or is extremely, extremely deconditioned. But even then, I would say that we have given CAR T cells successfully to people who have had organ transplants. We have given CAR T cells successfully to people who have congestive heart failure with a very low, ability for their heart to pump. We've given CAR T cells to people who have had prior strokes. We've given CAR T cells to people who are, I think, upward in their high 80s. And I would give CAR T to someone in their 90s. I just haven't had, it hasn't come up yet, but I would. And so to me, it's much more about the patient in front of me. And if there's someone who is relatively healthy and I think that they can tolerate it, there's no upper age limit. And even with a lot of the medical comorbidities, we can usually get them through with the help of other disease specialists. And so I would say it's actually on my end, it's the rare patient for whom we truly can't do CAR T due to medical comorbidities. Dr. Klein, I see you want to add.
Yeah, I think for me, if you can get up out of a chair, let's go. Let's do it. Especially since, the initial CAR T product that Sam mentioned, which is Axi-cel or Yescarta, that one gets a little spicy. But the third of the three that are approved, Breyanzi or Lisa-cel, I mean, we see, such a low incidence of severe side effects that, uh, it really has broadened our ability to offer this as you mentioned, potentially curative treatment to people who are older and who have other health problems. I recently had a couple of women in their mid 80s who flew through it, perhaps to my surprise. So I think we often get questions from referring doctors about, they think of CAR T cells as being CAR T cell therapy as being similar to stem cell transplantation, but it turns out that there are many people for whom I would never offer a stem cell transplant that would absolutely recommend CAR T cell therapy.
Yeah, I couldn't agree more. Couldn't agree more.
Thank you both for chiming in on that. And another question for you, Dr. Klein. If my treatment causes neuropathy, is that something that typically resolves once treatment is over?
That's the word on the street.
Yeah.
So yes and no. I think if you look at clinical trials that include drugs, including antibody drug conjugates like brentuximab vedotin, perhaps most notably, if you look at folks who are on those clinical trials, who are part of those clinical trials, um, and who develop neuropathy, the vast majority, if you look years later, of those people will report that their neuropathy is resolved entirely. A minority of people will report that their neuropathy is improved, but not totally resolved. Now, in clinical practice, I often tell folks that they will experience some neuropathy, very likely with some of the therapies they're receiving. And, if we're giving therapy with the intent to cure lymphoma, I'm willing to tolerate on behalf of the patient a bit more neuropathy than I would in drugs that we're using for neuropathy in a palliative setting. That being said, neuropathy often takes many months to resolve. And although it's not life-threatening, it's a significant impactor of quality of life. And so that's something I've realized with experience in this job, that neuropathy is what people will tell you about when they come back to see you a year later after their lymphoma therapy. Sometimes it's still bothersome. So in short, most often it resolves if it's recognized before it gets too severe and the drug is either reduced in its dose or omitted from the treatment. Then that can lead to a higher likelihood of the neuropathy improving or resolving. But there are some people who have more chronic symptoms after they go through lymphoma treatments.
Thank you, Dr. Klein. And I'm actually going to give you another question, Dr. Klein. Now or in the future, can antibody drug conjugates or bispecific antibodies be used to treat T-cell lymphoma?
Uh, yeah, T-cell lymphomas are tough because it turns out that, for example, with CAR T cells, there's bispecific antibodies, or even antibody drug conjugates that target B cells. People can live without B cells. They can live totally normal lives with no B cells, very low antibody levels. They're perhaps a bit more susceptible to infections. But the antibodies can be replaced with infusions of pooled antibodies. And so it's not such a big deal for most people. It turns out, though, when you get rid of malignant T cells in T-cell lymphomas, you're also targeting healthy T cells. And so people don't do well without T cells. They're really susceptible to odd atypical infections. And so we haven't found, in my way of thinking, a way around that just as of yet. Although there are people who are developing CAR T cells, for example, for T-cell lymphomas, they're not really ready for prime time yet. And so could it be in the future that these therapies are used? I suppose, but I don't think in the next couple of years, at least. I don't know, Sam, if you feel differently about that.
Yeah, I completely agree with that. I mean, what makes it tough is you need to find the target that's gonna kill the bad guys, but not the good guys. And that is surprisingly difficult, not just in lymphoma, but in general. And I think that's one of the major limitations of CAR T is in lymphoma, we have good, at least B-cell lymphoma, we have good targets. But actually in general, that finding the right target is very difficult. And so completely agree.
Thank you both. And kind of rounding out that question for Dr. Yamshan, we have a lot of interest in CAR T-cell therapy and as it's being researched for other types of lymphomas. So specifically, people are wondering about Waldenstrom and marginal zone lymphoma and what you might be seeing about those.
Yeah, and I think I saw a few questions in there in the chat also about follicular lymphoma too,
Yeah.
And mantle cell, because I think we mostly talked about for the aggressive lymphomas, but so for the indolent lymphomas, so follicular and mantle cell, those are FDA approved indications and are quite effective treatments in the right setting, and so we actually very frequently do follicular and mantle cell lymphoma CAR Ts. For marginal zone lymphoma, interestingly, so in the same trial that led to the initial approval for follicular lymphoma, marginal zone lymphoma was also included in that study. And so in Zuma, called the Zuma 5 study. And so there actually is precedent, and we know that it's effective in that setting, but it's not technically FDA approved. There are some investigational CAR Ts, and we actually sometimes are able to kind of get it off-label for marginal zone lymphoma in specific circumstances if like the pathologist says, oh, I saw a large cell in there, and then we're able to like kind of call it large cell lymphoma. But so we have treated several people here commercially with marginal zone lymphoma that we were either able to get off label or there was a question of transformation, and we've had quite successful results. Waldenstrom's is interesting, and because Waldenstrom's kind of sits in the middle between a couple of different diseases, and so there's actually a couple of different CAR T cell trials out there, but I think that they are they're not I don't think they're close to approvals in that setting. I do have one patient who had a transformed Waldenstrom's that I have treated with CAR T quite effectively. But so far I think it's still in the trial mode for Waldenstrom's.
Thank you both very much for answering all of these wonderful questions. And of course, we couldn't get to all of them today, especially kind of the more specific ones. But if you did want to be connected with someone from our helpline, all of that information to contact them is at lymphoma.org. But the helpline is, the email is helpline@lymphoma.org. And our number is 800-500-9976. You can also respond to the email you get as a follow-up to this program, and I'm happy to connect you with the helpline or with Dr. Klein or Dr. Yamshan if you have specific questions as a follow-up to today. And before we wrap up, I also want to remind you to please complete the program evaluation because we really value your feedback. Otherwise, thank you so much, Dr. Klein and Dr. Yamshan. I hope you have a great rest of your day.
Take care,
Thanks for
everybody.
having us.
Thanks for having us.
Thank you.